Excellent. Thanks so much for the introduction, and thanks to everybody for sticking around to the last presentation of this workshop. As a part of the organizing committee, I'm not sure how I ended up last. Probably could have had better foresight there, but I appreciate you all still paying attention. So yeah, today I'm going to be talking about advances in nucleic acid vaccine technology. So just to give you an outline of my talk, for those of you who aren't familiar with self-amplifying RNA, I'll talk a little bit about that in the current state of the art. I'll talk about some of the work we've done on comparing mRNA and saRNA expression in vivo, as well as some of the optimization that we've done on saRNA protein expression, as well as immunogenicity and understanding the dynamics there. So just to declare my conflicts of interest, I am a co-founder of a company called VaxEquity, which develops saRNA vaccines, so just keep in mind that I am very biased. So it's been a really exciting time to be in the field. I think as many of us in the room can attest to, the plane is finally off the ground as far as having approved mRNA vaccines, but what my lab at UBC is doing is thinking about where do we go from here. So the research that we do in my lab really kind of focuses on three key aspects of this technology. So first is the RNA molecular design. So how do we design RNA that expresses more proteins and is more stable? The biomaterials design, so how do we get this RNA to the cells and tissues where it needs to go, as well as thinking about how do we make better vaccines and use RNA for other applications. So other vaccines such as bacterial pathogens, understanding the interactions between RNA, the nanoparticles, and the immune system, as well as expressing monoclonal antibodies, which we had a really nice talk about earlier. So one of the priority areas in the field is minimizing the dose of RNA, so that we've seen a lot of really nice data from all of the clinical trials that have been performed in the context of COVID-19 vaccines. This is some data from one of Moderna's trials where they looked at 3 different doses of RNA, so 25, 100, and 250 micrograms, and this was just the phase 1 study where they were looking at side effects. And so you can see from this, they've put the side effects into 3 different categories, so mild, moderate, and severe, and I think what's very apparent from this data is that as you increase the dose of the RNA, you also increase the severity and the frequency of side effects. So this means there's a real need in the field to minimize the dose of RNA that's required for these RNA vaccines, especially when we're thinking about using them in the context of not a pandemic. So for example, for influenza vaccines, as well as for other applications of RNA, where those side effects may not be tolerated. So one of the ways that we can think about lowering the dose of RNA, there's many, but one of the approaches, which is an approach that my lab uses, is using self-amplifying RNA. So I have this structure here at the top of the slide of a typical messenger RNA, which I think we're very all familiar with. So it has a 5' cap and 3' poly A tail, as well as an untranslated region at both the 5' and 3' end, and then your coding sequence, which of course in the context of a vaccine is your antigen. For self-amplifying RNA, many of the structural components are very similar, so it has a 5' cap and a 3' poly A tail, as well as an untranslated region at either end. And then you'll notice there are these 4 NSPs, or non-structural proteins, which are derived from an alpha virus. And these proteins, once they're expressed in the cell, form a replicase complex, and I'll talk about the mechanism in a little bit, as well as you have your coding sequence for your antigen. So if we were to compare, this is a very simplified schematic of what happens when an mRNA or saRNA enters a cell. So first on the right, we have your typical messenger RNA. So once one of the molecules enters a cell, it engages with the ribosome, and then you get translation of your antigen. On the other hand, for the saRNA, it starts out very similarly. So once one of the molecules gets into a cell, it engages with that ribosome, and you do get some translation of your antigen, but you also get translation of that replicase complex. So that replicase then goes back to that original strand of RNA and makes copies of it. I have just 3 depicted here, but it's actually on the order of thousands within the cell. And so because of that, you get a much higher level of antigen translation. So because of this, there have been studies in the field that have shown that you need about 100 times lower dose of saRNA than mRNA, at least in mice. So one thing I wanted to highlight, which is obviously quite related to Deb's work as well, which is that the first siRNA vaccine was approved. This is a COVID-19 vaccine that was approved in India at a dose of 5 micrograms. So obviously a really exciting milestone in the field. One of the things that I wanted to start off by talking about is just looking at the difference of these expression profiles for messenger RNA versus saRNA in vivo. So there's actually very few published studies of this. This is actually unpublished data from my lab, where we formulated both mRNA and saRNA in the exact same lipid nanoparticle and administered them at a dose of 1 microgram. And then we monitored the luciferase expression over the course of a month. And what you can see here is that the mRNA, as we would expect, within 4 hours, we're seeing really high expression that tails off after about 4 days in the study. Whereas the saRNA starts to, you get kind of little blips of expression at the very beginning, but then you start to see it really take off around day 4 to 7. In this study, it peaks at around day 10 or 11. And then you see this slow tail off over the rest of the course of the month. And we still see a low but variable amount of expression in the mice after 30 days. So I think this really highlights one of the main differences, not only when we're thinking about using saRNA for vaccine purposes, but also for protein expression purposes, which is that you get that much longer duration of protein expression compared to mRNA. So one of the things that's really important, especially in the context of any RNA vaccine, but a therapeutic as well, is really thinking about how the RNA is sensed intracellularly. So obviously, this is from a review that Norbert published a few years ago now, which I reference in basically all of my talks. But I think it does a really nice job of explaining just really what we're thinking about, as well as where the gaps are. So we know that many of the sensing mechanisms for RNA within the cell, so those in the endosome, such as TLR3, 7, and 8, as well as cytosolic ones, some common ones such as MDA5 or RIG-I. One thing that really always stands out to me, which I think some progress has been made on, including Michaela's presentation this morning, is the carrier sensing and the big question mark next to it. So I think we're still really learning more about how the delivery vehicle actually is sensed by the cell, and this is no doubt an important aspect. For the messenger RNA vaccines, obviously we use most of the time modified nucleotides. We don't do this for saRNA because there's very little published data on this, but actually if you incorporate the modified nucleotides, this completely messes up the replication, and so you don't see expression from this. So that's at least with the traditional modified nucleotides. There are others out there looking at using different modified nucleotides for saRNA, but nobody's discovered really what works for that yet. So because of this, we really need alternative strategies to modulate the immunogenicity to saRNA in particular. So I don't think I have to convince anybody here, but humans are not mice, ferrets, or monkeys. I think this is one of the main challenges in the field, which is that we use all of these preclinical animal models to test our systems, and oftentimes things look amazing in animals and then really struggle once they get into humans. And so I always think back to, this is actually a study that was published in 2017 from Moderna. I'm still always surprised that their lawyers let it out the door, but I appreciate the transparency of it. So this is a study using a flu vaccine, and they looked at a really nice parallel study in mice, ferrets, monkeys, and humans. This is the HAI titer, which is the correlate of protection for flu vaccines, and in mice, and you can see that the initial findings in mice was the HAI titer was about 1,000. When they did it in ferrets, it was up to 10,000. So they did it in a non-human primate study, which was also at around 10,000. And so if you saw this preclinical data, you'd say, wow, this is going to be an amazing flu vaccine. Let's go into a human clinical trial. When they did it in their human clinical trial, they saw a much lower HAI titer, which the average was just below 100. And so really, obviously, a lot of progress has been made. This was published in 2017, and so I'm sure there's been many changes to the flu vaccine design since then. But really what I want to highlight is just the disparity between what they saw in every single other preclinical animal model and what they saw in humans, which I think really motivates us to think about the models that we're using to optimize and develop these systems. It's really easy to get things to work in mice, but much more difficult to get things to work in humans. So because of this, one of the projects that I was working on towards the end of my postdoc is really thinking about how we can modulate the immunity against saRNA. So in order to do this, we screened a library of 10 different interferon inhibiting proteins. So these are proteins that viruses have evolved to use to modulate their own interferon activity. And so we thought we could repurpose this for saRNA launched vaccines. So at the top, you see we have our wild type replicon with the structure as I described before. And on the bottom, all we did was insert these 10 different interferon inhibiting proteins, or IIPs as I call them, separated from our gene of interest with a T2A cleavage site. So this guarantees that this protein is being expressed in the same cell where our saRNA is replicating. So the first thing we wanted to do was just test these and see if we saw a difference in protein expression in vitro in a variety of different human cell lines. So we used 3 different cell lines, and I'll kind of walk you through why this was. There's a lot of data here, so let me orient you. So the black bar is our wild type replicon. On the y-axis, we have our RLU, which is the luciferase expression that we were looking at, and then we have our library of 10 different IIPs and 2 different batches of those to see if there was just any variability that we were seeing between the 2 different batches of RNA. So the first thing we wanted to do was to look in HEK cells. These cells are known to not have a complete interferon pathway, so we wouldn't really expect to see a difference between the wild type and any of our IIPs, which indeed was the case. They all had very similar levels of expression. However, when we looked in both HeLa and MRC5 cells, which are known to have not perfect, but a more complete interferon pathway, we see that there are 2 standouts that had much higher protein expression. So one of them was an interferon inhibiting protein from PIV5, and the other is actually an open reading frame from MERS. So based off of this data, we then went to test this in a number of different systems. So the first was to look in mice. And to be honest, we didn't really expect to see a huge difference in mice because mice are so much more tolerant of the saRNA than any human system is. But one of the things we can always look at is the dose dependence and nonlinearity. So I'm sure many of you have seen, if you've ever done a dose finding study, that there always becomes a point where giving more RNA does not result in more protein expression. And that's exactly what we're seeing here. So on the y-axis, we have the total flux. So this is the amount of luciferase expression that we saw. We imaged the mice at either 7 or 10 days at this, in this case. So this is kind of the peak of the protein expression we see in vivo. We used 3 different doses, so 0.2, 2, and 20 micrograms. 20 micrograms is a very high dose of saRNA where we start to usually see detrimental effects of the dose. And that's exactly what you can see here. So with our wild type fluke, which is in black, you can see at that 7-day time point. At the 0.2 micrograms, we see okay expression. At the 2 micrograms, it's much higher. And then when you go to 20 micrograms, there's barely any visible protein expression anymore. So what we see is when we incorporate this interferon inhibiting protein that we partially abate this dose dependence. So it's very similar to our wild type at the 0.2 dose. At 2, it's slightly higher. But the real difference we see is that when you dose at 20 micrograms, actually the majority of the mice, you still see protein expression. It's lower than the 2 microgram dose, but it's not completely gone. And so what this is telling us is that this is actually playing a role in the protein expression that you're seeing, but also the dose that you can give. So we went on to test this in a system that's a much better screen for us. So what we did to do this was to look actually in human skin explants. So we get these from local plastic surgeons, and it is pretty disgusting. These are just skin that are being discarded after abdominoplasties or mastectomy surgeries, but it's actually a great research tool for us because it allows us to test our RNA in a human system with a native architecture, a variety of cells, and a complete human interferon pathway. And so what we did in this case is we were using saRNA that was encoding GFP as a reporter protein, and we included in that same construct our 2 different IIPs, so from PIV5 and MERS, and we looked at the number of GFP positive cells in these human skin explants, again at the same doses that we used in mice, so 0.2, 2, and 20 micrograms. At the time when this was taken, we then digest the tissues and look at the percentage of GFP positive cells, and so you can see at our lowest dose dose, there's really no difference. About 10% of the cells were GFP positive at the 0.2 dose. It goes up slightly to the 2 microgram dose, but really the main difference you see here is that with our wild type construct, when you go up to 20 micrograms, again, we see a lower number of cells expressing the protein, but when you have either of these interferon inhibiting proteins in it, we actually see an increase between the 2 and 20 microgram dose. So this is, these results were much more profound than what we saw in mice, which also clues us into that it's actually very important to optimize these in a human system, because what you see in mice is a much more muted effect because it just doesn't make as big of a difference. So the next thing we went on to do is to test how incorporating these IIPs into a vaccine impacts the immunogenicity. So in this case, we used a rabies vaccine as a model antigen, and we tested this in rabbits. We also tested this in a parallel study in mice and rats, and we didn't see any difference in the immunogenicity, again, likely because these things work very well in mice and rats. But rabbits are a little more interferon sensitive, and so we thought this would be a good model to test it in. So in this study, we gave the animals 2 doses, so 0 and 4 weeks, and then we looked at both the antibody titers, which is on the left, and the neutralization, which is on the right, at 0, 4, and 6 weeks. So you can see with our wild type that after 4 weeks, we do see antibody titers, which are just very slightly boosted after 6 weeks. However, with our interferon inhibiting protein, there is a slightly higher titer after 4 weeks and a significantly higher titer after the 6 weeks, so after the boost. And this, these findings are just directly reflected in the neutralization assay as well. So this tells us that not only are we getting kind of more protein expression in a human system, but we also see it improving the antibody titers in a more interferon competent preclinical animal model. So in conclusion, I just wanted to kind of go over this. So for the siRNA, we see that it results in about 10 times longer protein expression in vivo compared to messenger RNA. We found that these 2 interferon inhibiting proteins, PIV5 and ORF4A, are able to abate this nonlinear dose response in a human tissue, and that they result in higher protein or immunogenicity in a preclinical rabbit model, but not mice or rats. So I just wanted to think about how this is applicable as we think about going forward. So this is actually just taken directly from BeyondTech's website, but I think is a really interesting view of the field, which is that, today we have these RNA vaccines, but thinking about modulating the interferon response is really important for all of the therapies and other applications of RNA going forward. So, we have the modified nucleotides, but I think there's lots of rooms for other technology and other approaches for modulating the response of RNA, which is imperative for therapies. So you'll note that some of the things they're working on now that you could imagine being treatment or a therapy in the near future is mRNA infectious disease vaccines, cancer vaccines, and CAR-T amplifying vaccines. But in the future, they're also considering applications in autoimmune diseases, allergy, inflammation, and regenerative medicine. So I think there's lots of different applications that are out there. So really exciting time to be in the field, but also really exciting time to think about how we can make these systems even better. So with that, I'd like to acknowledge everybody on my team who contributed to my work, my current lab members, as well as previous collaborators and supervisors at Imperial College London, as well as our funding sources. I did want to give a quick plug. So we are starting an RNA and formulation core at UBC. This is being done in collaboration with Peter Cullis and Eric Jan. So the point of this is to make RNA and formulations more accessible to people in the field. So what we're aiming to do is to offer services for constructing and designing custom plasmids, synthesizing all different types of RNA, then doing the purification and quality control, as well as formulation into lipid nanoparticles. We have a director who's starting July 1st, so hoping to do some beta testing shortly thereafter. So please feel free to contact me if you're interested. And I'd also like to point out that we're aiming to do this for both academic and industrial users. So I know one of the hardest things in the field right now is just where do you get really good high quality RNA and why is it so expensive? So we're aiming to change that for everybody. And with that, I will take any questions.